higher secondary school Langate. I continue the topic, that is the detailed account on Monira, and in that aspect I am teaching you the best suitable representative of the group, that is E. coli, because it has all features it must be present in the bacteria. All the bacteria do not possess all the features. Some are simple, leading from simple to complex. Several differences arise, and as such we must treat that bacteria which contain all features. And in that respect we have chosen E. coli, Escherichia coli, mostly found in the gut of human gut, and also in rudimentous, that's the digestive tract of these animals. Ultrastructure, in that respect I have taught you the first outermost appendage of the bacteria, that's called flagella, and I also told you the background, the morphological background of the bacteria. Today we will continue the same topic, and the second teaching you the structure of the structures of the ultrastructural appendages or the ultrastructural components of the bacteria. In that respect, today I will teach you bacterial pili, also called family. A special kind of proteination or family, pelli or family. Pelli is plural, pelli or family. These are special kind of proteinaceous appendages or projections. These are special kind of proteinaceous appendages or projections present on the outer wall or the cover the bacterial cell. There is a covering of the bacterial cell and a special kind of proteinaceous appendage that's called pellet. It's called pellet. These pellets are smallest ranging. If we talk about these pellets, are formed of special kind of protein monomers. Special protein monomers. Protein monomer means protein molecule. Molecules which form a bigger, smaller molecules form bigger molecule. That is, bigger molecule called polymer, and the units of polymer are called monomers. So the special protein monomer in that is called pellet. It is called pellet. Pellet had once been put in entrance exam, and with this neat entrance exam, the pellet protein is present in what kind of the flagella. Then the options were shown, that's flagella, cilia, pellet, etc. So the pellet is present in the projection, that is the pellet or family, which is a protein monomer, and these pelli are present all over the body, all over the body surface, and covering the whole body surface. What they help, these pelli help bacteria in sensitizing or to find food, help the bacteria to find food. But the longer pelli, if present, they are considered as sex pelli. They help in recognizing the bacteria which are ground. There are two kinds of bacteria: bacteria positive, bacteria gram positive, bacteria gram positive, gram negative. Or there are bacteria which are differ in. Some bacteria do positive. I will tell you lysosomes. In later proceeding further, I will tell you lysosomes. Some bacteria have extra nuclear chromosomes. Some do not have. A small fragment of DNA is present in some bacteria, and some bacteria lack that small fragment of DNA. One which lack are called negative. Those do not lack are called positive. Then we have two kinds of bacteria: Positive means they contain the extranuclear DNA, and those who do not contain, they are negative. I will tell you later. So asexually helps bacteria to adhere together during conjugation. They help bacteria to come close and transfer genetic material. So whereas the pellet is concerned, the pellet protein, the monomers, or they are arranged in such a way, they are arranged helically, enclosing the space. They are enclosing the space of 20 angstroms, 20 angstrom space, and this length of the helen is length of pellet is about 0.5 millimeter, and its diameter is said to be about 70 to 100 angstroms, 70 to 100 angstroms. And this completes the belly or fabric. The second important, very important, the structural feature of bacteria is so important. Not only in this bacteria, other bacteria as well. I have given this bacteria an example. Other bacteria, and there is another, that is adherents. First of all, I will show I will call here. Adherents are, these are the polysaccharides. Adherents are polysaccharides, or glucose units. When glucose units are held together, these are present outside the cell wall. Polysaccharide units, or polysaccharides present outside the cell wall, of bacteria. That is, this is bacterial cell. It has this bacterial cell is cell wall, and outside the cell wall is present a thick layer of glucose, or thick layer of polysaccharide. Glucose units, when combined together, they form polysaccharide. Polysaccharides are various kinds. For example, structure of polysaccharides, for example, storage parasaccharides. You will read these things in higher classes. And the polysaccharide present outside this cell wall. And in bacteria anthracis, in one of the bacteria anthracis, it is present singly, that is, the polysaccharide is present without any mixing. But in others, this polysaccharide is mixed with proteins. It is mixed with protein. When it is mixed with proteins, form the capsule. It's called capsule. And depending upon the thickness of the capsule, there are two kinds of capsules. It may be a microcapsule, or it may be a microcapsule. Micro. These are a polysaccharide, or the adherents, structure present outside cell wall. It helps bacteria to tire out the host conditions, tire out the hot conditions. Whenever the conditions become unfavorable, it helps bacteria to survive. So there is another important structural feature of the bacteria, that is very, very important feature of bacteria, which separates bacteria from the other unicellular microorganisms. One of the features, not all, one of the features of the bacteria which separate it from this protozoans, from this protista, that is cell wall. In addition to the bacteria do not possess the membrane-bound organelle, it do not, but they possess cell wall. Fourth important feature is cell wall. This is the first outer covering of the bacteria, first outer layer of bacteria formed of cellulose. Bacteria. So far, this bacteria, it has outside, has a cell wall, and outside to the cell wall, I told you, is present polysaccharide, that's called polysaccharide, which may be simply a polysaccharide or a capsule. But outside, inside to the covering, or the most important structural feature of the bacterial cell is cell wall. This is cell wall, and this cell wall in bacteria is also called it is called the peptidoglycan. It is peptido. It has several names: peptidoglycan, glycopeptide, glucopeptide, murine. It is also called murine. These are same names: peptidoglycan, murine, glucopeptide. These are the names for this cell wall structure, cell wall composition, and this peptidoglycan, glucopeptide, murine. It is composed of two kinds of amino sugars. That is N-acetyl. This is N-acetyl. We are going to S2s, how the S2 derivative of N-acetyl glucose amine. And N-acetyl muramic acid. These glucose amino sugars are present. For example, if this is one of the amino sugar, it is this one of the amino sugar. This is another amino sugar. They have alternately in this way. They are present, and there is another amino sugar. It is difficult to explain things clearly here because time shortage, and also keeping in view your net package. This is at the fourth position of this first position of this, that is carbon fourth of this, carbon first of this, the linked together. So linkage is 1 to 4. 1 to 4 of 4. One linkage you can, if you write this is 1, then this is 4. 1 4 linkage, and these units are then the units are then held up or linked together by protein units, protein molecules. That is, this is a protein molecule, this is a protein molecule, this is a protein molecule. Similarly, on this side. Similarly, if there is another sugars on this side, so this forms a network. This together forms a network. The sugar molecules, amino sugar molecules will form backbone, and these backbones together, when they are connected or linked by protein molecules, they form a network, and this network forms a thick wall around a bacterial cell. And this thick wall around bacterial cell, there are what kind of thickness it has. Bacteria on this basis of thickness are divided as gram-positive and gram-negative bacteria. You will read it in higher classes. And simply keep in mind that bacterial cell wall is composed of two kinds of amino sugars, N-acetyl glucosamine A and N-acetyl muramic acid, and these are held together in 1 to 4 linkage, and once these backbones are formed, they are linked together by amino acids. 4 kinds of amino acids: L-alanine, L-D-alanine, lysine, and also uramic acid. Glutamic acid, glutamic acid. These are the amino acids present in the cell of bacterial cell. This completes your today's topic because its time is sufficient. Time is not so sufficient. I could go further, but it is best to save your time, save my time, also to keep in mind the loading of the video. Thank you very much. Also, if I will try, if in the later time, if I will find time